Swayam Prabha. Digital India. Educated India.
Hello students, welcome to the 7th lecture of this module, which is Nanotechnology in Water Treatment. So in this lecture, you will be learning about kinetics of toxic dye removal, experimental data fitting, removal of pesticides, pharmaceuticals and microbial contaminants. So industrialization has led to pollution of water bodies by releasing dyes, pesticides and pharmaceuticals into water bodies. Okay, so water basically is very vital for our life. As you know that major part of our cells is also made up of water. So water is so essential that whenever we are looking for life on other planets also, we are looking for water only. So you know how essential water is for the propagation of life. So but the current situation is, you already know that water is getting polluted from everywhere, from industries to vehicles to cars release oil drops, and you know, through water cycle all these pollutants and contaminants, they get recycled into rivers and seas, and finally they reach seas and they travel through vast distances. So the pollutants get spread all over the world through you know, water keeps moving and it gets spread all over the world. So it has created a lot of menace, and that needs collective efforts for remediation. And microbial contamination is also a huge threat to public health these days, as you already know about this brain eating amoeba these days being talked about. So you know how threatening microbial contamination of water bodies is also these days. So you know how threatening microbial contamination is to us. Then conventional treatment methods are not enough. So nanotechnology basically is offering efficient and scalable solutions today. So how does it do that? So why nanotechnology is being used for water treatment? So as you already know by now that nanomaterials have a high surface area and reactivity of, as you already know that nanomaterials have a high surface area and reactivity. They have enhanced absorption and catalytic degradation power, and they can selectively interact with pollutants, and their compatibility with biological systems is very high. Okay. So nanotechnology has emerged as a promising method for water remediation. The nanomaterials being currently used for water treatment are basically metals and metal oxides of nanoparticles, and carbon-based nanomaterials. These can be carbon nanotubes and graphene, and these can be polymeric and hybrid nanocomposites, or bio-inspired and functional nanomaterials. So these are some specific nanomaterials that are currently being used for water remediation methods. So aquaporin based biomimetic membranes. These are used as for low pressure desalination. The properties are low mechanical strength, strong permeability for ions, more excellent water permeability and superior selectivity. So these are some nanomaterials that are used for water remediation methods. So carbon nanotubes and membranes. Applications are desalination, removal of highly biodegradable pollutants like pharmaceuticals and antibiotics, and the advantage is that, and not advantage, carbon nanotubes basically they can be costly, they can pose certain health risks, and although they have a good sorption efficiency, energy efficiency and fouling abilities, and chlorine resistant. So there can be advantages as well as disadvantages for this. Then there can be magnetic nanoparticles which are used for advanced wastewater treatment and disinfection, that is removal of arsenic or elimination of microorganisms. So properties are magnetic separation and redox behavior. Okay, so the advantage of using magnetic nanoparticles is that they can be removed via magnetic fields. after being utilized, and they can also be recycled again. So nanocomposite membranes, these include reverse osmosis or the elimination of micropollutants. The properties include fouling resistance, boosting permeability, and it is highly selective and antibacterial. Then nanofiber membranes. These can be used for water treatment, standalone filtration device. These are portable filtration devices. These include nanofiber membranes, which filter out some pollutants and microbes. Then filter cartridge and ultrafiltration. Properties include high pressure efficiency, custom fit, high porosity, and bactericidal. Then there are nanofiltration membranes, which can reduce virus and bacteria from water, heavy metals, hardness, and odor. So, the properties are high selectivity, relatively low pressure, charge-based rejection, and minimum blockage, concentration polarization. Okay, so these have minimum blockage. Otherwise, whatever membranes you use, there is a case of fouling. So, nanozeolites, disinfection processes. These are basically used for disinfection processes. Nanoscale zeolite particles immobilized in controlled release of antibacterial disinfectant agent. So, as you know, these are highly porous materials. These can be immobilized in a controlled release of, you can put some antibacterial disinfectant agent into you can integrate some agents into it, and that will result in a controlled release of these agents. Then self-assembling membranes. These are used for ultrafiltration. As you know what ultrafiltration is, that is filtration through a semi-permeable membrane where microbes are left behind and water and salts pass through. So properties are, these are effective in small amounts. Okay, even these are effective even in small amounts. Self-assembled functional membranes and nanomembranes, these are, and ceramic membranes on laboratory scales. Basically, ceramic membranes are being used. Then nanoscale zero valent iron particles. I'll be telling you what these are. Zero valent iron particles. These can be used for the treatment and remediation of wastewater, that is chlorinated solvents, dyes, pesticides, etc. Then properties include high reactivity and stability, surface modification. Then silver and titanium dioxide and nanoparticles. The applications are anti-fouling surfaces. It is used for creating anti-fouling surfaces. It is used for antimicrobial activity and decomposition of organic compounds. So properties include, basically it is antimicrobial and highly reactive, and it is highly stable also, and it is also very durable, but it's a bit toxic. So nanomaterials like polymeric nanoadsorbents, these are dendrimers. All these structures, if you remember, I've already told you in the structures in the classification part of nanoparticles. So these result in the elimination of heavy metals and organics, and properties include high mechanical strength, electrostatic interaction, the unique hierarchical structure of these, and antibacterial agents, and absorb. These are highly antibacterial and they also absorb heavy metals. So let's first start with toxic dye removal. So what are the pollutants that basically pollute water bodies? These can be either toxic dyes, these can be pesticides, or antibiotics that are released in water. These are very toxic to human health, and so it is important to remove these from the water bodies. So we are targeting these one by one. So now let's start with toxic dye removal first. So textile and dye industries, these release persistent dyes. Okay, so these constantly keep releasing dyes that these percolate through ground also and get into water, and these go through rivers, and the effluents, they get released into water bodies and pollute the entire water system. So these textile and dye industries release persistent dyes. Then nanoadsorbents capture dye molecules efficiently. So these nanomaterials can also act as nanoadsorbents for capturing some dye molecules. nanocatalyst can enable oxidative degradation of these dyes. And what nanomaterials are doing, they are basically increasing the removal efficiency, and they are speeding up the reaction. That's the figure, and you can see that is textile effluent, and these are the pollutant species. Okay, so that's your water body, that's the textile effluent coming into water bodies, and these red dots are basically pollutant species. So now you're putting a mixture of hydrogen peroxide and ferrous salt into it. So what is happening is that via Fenton reaction, basically this hydrogen peroxide is oxidizing Fe2+ to Fe3+ and creating this hydroxyl species. Okay, hydroxyl anion and hydroxyl radical. So these hydroxyl radicals are going to break down your pollutant species and break them down into some harmless products. Either CO2, H2O, these will be breaking down them into harmless products and clearing the water. That is iron oxide sludge. That's what the famous Fenton reaction is. So basically you put hydrogen peroxide. Hydrogen peroxide acts as a fuel. Okay, so these are your iron. You are putting, let's say, you are putting some iron oxide nanoparticles, or you can also put iron doped carbon dots into it. So basically you are generating some iron nanoparticles, iron oxide nanoparticles. Now your hydrogen peroxide, that is the fuel. It is what it is going to do is that it's going to oxidize this into Fe2+ to Fe3+, and a hydroxyl and hydroxyl radical. Then this H2O is also going to reduce Fe3+ to Fe2+ and release H+ and hydroperoxyl. Okay, so all these are reactive oxygen species that are going to break down your organic pollutants very efficiently. So this hydroxyl radical also reacts with hydrogen peroxide to create a superoxide anion. Now this superoxide anion will react with Fe3+ to produce Fe2+ and O2. And this O2 and this Fe2+ is going to be recycled back to here. This Fe2+ that is being generated over here will get recycled back to here. And this is how the entire cycle goes on to clear your water bodies by breaking down these reactive species that are being released, are constantly breaking down your pesticides or toxic dyes that we are talking about, your organic compounds, into some harmless products. So that's a zero valent iron nanoparticle core shell model. Okay, so what you can see is that there's a zero valent iron over here, and it is covered, the exterior is primarily made up of iron oxides and hydroxides. So that is the exterior, which is made up of iron oxide and hydroxides, and the interior is a zero valent iron nanoparticle. So while the center comprises of metallic iron, this is metallic iron, and it is surrounded by reactive iron oxide and hydroxides. So iron nanoparticles as a consequence display characteristics of both iron compounds and metallic iron. That's an example of the toxic dye removal, the Congo red dye, and it is being removed by copper ferrite nanocomposite. So what happens is that via UV radiation, as you can see, this is getting activated. Copper ferrite nanocomposite is getting activated, and the electron, you will see it will get excited and jump from the valence band to the conduction band, creating a hole pair over here and electron over here. So a negative charge over here, a positive hole over here. So what this is doing is that this is releasing OH radical from H2O2, and this is releasing OH radical from this is releasing OH anion from H2O. So what these radicals are doing? These radicals are breaking down your toxic dye into non-toxic products, that is carbon dioxide and water. So that's before and after picture. Like this is the Congo red water polluted with your Congo red dye, and this is your clean water. So that's the experimental data, and you can see how effectively this works. Then that's the magnetization curve of reduced copper ferrite. So the advantage of using copper ferrite is also that it is also magnetic. So it will be highly magnetic, and you can also remove your nanomaterials from the water after their treatment. So you can remove that and reuse that. That's the magnetization curve, and you can see a narrow hysteresis loop. This ensures that the catalyst could be easily separated by a magnet and reused from the aqueous solution. That's a heterogeneous Fenton-like degradation of malachite green dye using core shell iron oxide SiO2 nanocatalyst. So that's your iron oxide core, which is surrounded by SiO2 core shell, and this basically, what is it doing? It is basically degrading your Mg dye via Fenton-like reaction. The same reaction that I've already explained before, it is generating hydroxyl radicals, which are highly reactive species, and these are going to break down your Mg dye into degradation products via the redox cycling of your Fe2 plus to Fe3 plus to Fe2 plus. That's the redox cycling that is happening over here. That's the functioning of a micro nanobot. So I'll be explaining these in my next lecture, where I'll be telling you what these bots actually are. So basically, what do they do? These are basically chemically propelled nano microbots. These also work on the same principle of Fenton reaction. What they are doing is they are degrading your pollutant, and that's polluted water, and pollutant absorption is taking place. And heavy metals also, they sense heavy metals, and they remove that also. And these are the oxygen bubbles that are being released. These basically help in the propulsion of this bots. These micro nanobots can either be spherical or they can be Janus particles. They can be asymmetrical. Half of it is made up of some other substance, half from the other. That can be used to integrate properties as well as to create asymmetry for better movement of these bots in water. So that's the oxygen bubbles being released, and that's the direction of propulsion. Rate kinetics of dye removal. So this will describe the time dependent removal in the dye concentration. So whenever using a nanomaterial, what is happening is that for dye removal, that the dye concentration is decreasing with time. So degradation is happening continuously, and with time the dye concentration seems to decrease. So what is the kinetics behind this? What is being calculated is that pseudo first order kinetic model is basically applied to this. This reaction is following the pseudo first order kinetic model, and where rate constant reflects the removal efficiency, and it is important for the process optimization. So the nanocatalysts that are used for the rate kinetics, these are TiO2, zinc oxide, iron oxide, silver or gold nanoparticles, and doped metal oxides that are widely used for photocatalytic and catalytic degradation of dyes. Due to their high surface area, as I've already told you, they have a very high surface area, abundant active sites, which is because of the high surface area, the quantum effects, and enhanced redox capability. So understanding the kinetics helps us to evaluate their efficiency and optimize the operating conditions. So if you understand the kinetics behind every particular nanocatalyst, you will know, it will be easier to know their efficiency and to optimize their operating conditions. What is happening is that when a dye solution containing a nanocatalyst is being illuminated or irradiated by light, either UV or visible light, so it is being activated. The catalyst gets activated when light passes through it and degradation process starts. So that degradation process basically proceeds progressively with time. So as time passes, degradation continues. So the typical trend is that initially the adsorption equilibrium established on the catalyst surface. So after activation, what happens is that the concentration of a dye starts to decrease continuously. So over time, the dye molecules break into smaller intermediates and finally mineralize into some non-toxic products like carbon dioxide and H2O and some other harmless ions. So this is what happens. At first, the adsorption equilibrium is getting established on the catalyst surface. Okay. Then after the activation, the concentration of dye starts to decrease continuously. Then over time, the dye molecules break into smaller intermediates and finally mineralize into carbon dioxide and water and some other harmless ions. How do you experimentally monitor this entire process? The dye degradation process is monitored by UV visible spectroscopy. So I've told you in the characterization technique, how does this work? The absorption decreases at a characteristic lambda max that is characteristic to whatever dye that is being considered. So let's say your water is being polluted, you know that the water is polluted with the methylene blue dye. Okay, so the adsorption, the lambda max for it is 664 nanometer. So for that nanometer on that particular wavelength, you are going to study that. So what will happen is that you are experimentally monitoring it, and if your catalyst is working and your dye is being degraded, so you will notice that the absorbance goes on decreasing with time. That is how you monitor this. The expression, if C0 is the initial dye concentration and Ct is concentration at time t. Okay, so you've taken a water contaminated with a particular dye. So you take C0 as the initial dye concentration and Ct, like after adding catalyst, Ct is the concentration at any particular time t. Okay, let's say 5 minutes, 10 minutes, 1 hour, 2 hour and 16 hours. You continue you'll see that the dye is being degraded progressively with time. So Ct is basically the concentration at any time t. So you are calculating the concentration at particular time t. So degradation percentage is C0 minus Ct upon C0 into 100. That is the degradation percentage. That's the initial dye concentration. That is the concentration at time t. Or you can also use the absorbance formula, that is from the Beer-Lambert law, taking into consideration that absorbance is proportional to concentration. So degradation percentage will be A0 minus AT upon A0 into 100. That is the corresponding absorbance. That is the absorbance of the initial dye concentration, and AT is the absorbance of the concentration of the dye at that particular time t. Pseudo first order kinetics. So most dye degradation systems follow a pseudo first order behavior, especially when dye concentration is low. So dye concentration is low and it is very toxic at low concentrations also. So it is usually low and catalyst concentration is constant, and there is an excess oxidant, that is H2O2, dissolved oxygen is present. So this assumption simplifies the reaction to depend only on dye concentration. That's the rate law. dC by dt is equals to minus Kc, and integrating that you get this or this, and when you plot that, y is equals to mx plus c. Okay, y is equals to mx plus c. So that's your slope. That's your slope. If you are assuming a straight line, then this confirms pseudo first order kinetics, and this linear fit validates the kinetic behavior and the catalyst performance. So when dye concentration is low, pseudo first order reaction, that reaction mechanism is being followed. Experimental data fitting. So let's say you're performing an experiment, you've taken some crude water being contaminated with a dye. So now what do? That you take out the concentration versus time experimental data, and then you make a plot. Linear or non-linear kinetic fitting is performed. Okay, so extraction of rate constants and half-life. You take out the rate constant of the reaction and the half-life, and then this validates the adsorption or degradation mechanism of that particular nanomaterial that you are being using. So that validates the adsorption and degradation mechanism of that particular nanocatalyst that you are using for your degradation of that dye. Rate constant K, which quantifies how fast degradation occurs. These are some points to note. The rate constant, what is the rate constant? Rate constant quantifies how fast your degradation process is happening. Then units, for a pseudo first order, the unit of K is per minute or per second. Then interpretation, higher K means faster degradation, lower K means inefficient catalyst or unfavorable conditions. Okay. So that was the rate constant. So higher value of the rate constant means faster is the rate of the reaction, that is faster the degradation of the dye, and lower K means inefficient catalyst. The degradation process is basically slower and your catalyst is either inefficient or the conditions are not very favorable for your reaction. Then half-life is the time required to degrade 50% of the dye. So half-life is equals to 0.693 by K. Shorter half-life indicates superior catalytic efficiency. Shorter half-life will indicate a superior catalytic efficiency. As you know that higher K means faster degradation and lower K means inefficient catalyst. Higher K is faster degradation, okay. So higher the value of K, shorter is the half-life. So faster degradation and means shorter half-life of the dye that you are trying to remove. So it is degrading faster. When your half-life is less, it means that half of the dye has degraded very, it is degrading very faster. So the half-life is very less. So what it means is that in a very short time span, your half of the dye has been degraded. So that is what is meant by when you say that the half-life of a dye is shorter. Factors affecting the rate constant. These nanocatalyst properties can also affect rate constant, or operational parameters can also affect your rate constant. So your rate constant is affected by the particle size, the surface area, the band gap, the doping and the crystallinity. So these are the nanocatalyst properties that will affect your rate of the reaction. So as you already know, smaller the particle size, which means higher surface area already. So smaller particle size and also higher surface area, which will mean that the band gap is higher. If you remember my previous lectures, I have already told you this. So smaller the particle size, more will be the surface area and band gap, and all these factors will make it a better catalyst, as the absorption capacity will be more because of the higher surface area. More it will be able to absorb more dye molecules, and then it will act as a better catalyst. So doping also helps in functionalization and all. So crystallinity also, all these factors basically they affect the rate constant. Then the rate constant also depends on the operational parameters like the initial dye concentration, whatever dye concentration you are beginning with, that also affects the rate constant because that also determines your order of the reaction. And then catalyst loading or light intensity and wavelength, pH, temperature and presence of oxidants. These also factors affect the rate constant of the reaction. Then adsorption and kinetic models. The Langmuir-Hinchelwood model is the model for surface reactions. Pseudo second order model for adsorption control processes. We will be studying in this the pseudo second order model, and then correlation between adsorption capacity and kinetics, and these help to compare different nanomaterials. So in some systems what happens is that reactions deviate from the simple pseudo first order reaction and follow Langmuir-Hinchelwood model. So which accounts for the adsorption of dye on the catalyst surface. So what you are assuming is that your dye is not just floating in the solution, the adsorption is happening on the catalyst surface. So when you are considering your rate of reaction, you are assuming only that portion of the dye that is adsorbed on the catalytic surface. Okay, so you are not considering the dye that is in water, you are considering the dye that is on the catalyst surface. So now you are following the Langmuir-Hinchelwood model. So That's the expression for it. R is KKC by 1 by Kc, where capital K is the adsorption constant, small k is the reaction rate constant. So at low concentration, it behaves like a pseudo first order. That's Langmuir-Hinshelwood model, LH model for surface reactions. So what is happening is that the LH model is widely used to describe heterogeneous catalytic reactions, especially when photocatalytic dye degradation using nanomaterials such as TiO2, zinc oxide, iron oxide, or silver based, and some nano composites are being used. So what is the concept? The reaction occurs on the catalyst surface, not in the bulk solution. So this reaction is occurring on the catalyst surface. You are considering that whatever reaction is happening on the catalyst surface, the pollutant must first adsorb onto the catalyst surface. The reaction proceeds only when adsorbed molecules react with reactive species. So your reaction is going to proceed when the adsorbed molecules react with the reactive species. So pollutants then desorb, freeing the active sites. So this is what is happening. As assumptions is that the catalyst surface has finite and uniform sites. So your catalyst surface is going to have finite sites for the dye adsorption. So it will have, the adsorption will also follow the Langmuir monolayer adsorption. So it will be, the adsorption process will be in single layer around your nanocatalyst. Surface reaction step determines the overall rate. So your overall rate of the reaction is determined by the surface reaction step, when on the surface of the nanocatalyst your dye is getting adsorbed. Thus LH explains why photocatalysis often shifts from first order to zero order depending on the pollutant concentration. So depending on the pollutant concentration, your reaction order shifts from first order to zero order. That's the rate expression for LH, rate equation KKC by 1 plus Kc, where that's the reaction rate, C is the pollutant concentration, capital K is the adsorption equilibrium constant, and small k is the surface reaction rate constant. So what happens is that at low concentration, the value of Kc is very less than 1. So at very low concentration of the pollutant, at very low concentration of the pollutant, the value of that is Kc, basically that is K with a subscript C, basically that is the adsorption equilibrium constant. So at low concentration of the dye, the value of K will be very less than 1. So your reaction rate depends on K, so that will follow the pseudo first order reaction. Then at a high concentration, the value of Kc is very higher than 1. So in this case, your rate of reaction will be equivalent to small k, that is the surface reaction rate constant. So your order of the reaction will be zero order. Pseudo second order model or adsorption control. This model is applied when the adsorption is the rate controlling step rather than the surface reaction. So now at first you were assuming that you are not considering you were not considering the dye molecules in the water. You were considering only the molecules that were being adsorbed on the surface. And now this model is applied when the rate controlling step is basically, the adsorption is the rate controlling step rather than the surface reaction. So this assumes that the rate depends on the square of available adsorption sites, and adsorption is driven by chemisorption, which is electron sharing or transfer, rather than weak physisorption. So here adsorption is driven by chemisorption process. So this is common for carbon nanomaterials, metal oxides, and polymer nanocomposites. That's the rate equation. dQt by dt is K2 QE minus QT whole square. So integrating that, you get this, where QT is the adsorption capacity at time t, QE is the equilibrium adsorption capacity, and K2 is the pseudo second order constant. So the characteristics are that this provides an excellent fit for adsorption driven dye degradation. Predicts an equilibrium adsorption precisely, and this indicates chemisorption dominance. So a linear plot of T versus T by QT versus T confirms that your reaction order is basically pseudo second order kinetics. Your reaction is following the pseudo second order kinetics. So your adsorption capacity is correlated with the kinetics of the reaction. Adsorption and kinetics are not basically, they are not independent factors, they strongly influence each other. So higher the surface area and porosity means higher adsorption, and which means the faster degradation. So stronger stronger the adsorbent and pollutant interaction, and higher will be the rate constant. So a good adsorption will enhance the reactant availability on the surface, which will accelerate catalytic degradation. So that is what you can conclude, that a good adsorption will enhance the reactant availability on the surface. So on the surface of your nano catalyst, more will be the smaller the particle will be, and more will be the surface area, and a good adsorption means these are your, let's say, dye molecules being adsorbed on the surface. So a good adsorption, what will it do? It will enhance the reactant availability on the surface. So as you can see, more number of sites, active sites, will be interacting with your pollutant molecules. So a good adsorption will result in the acceleration of the catalytic degradation process. So the practical meaning is that materials with a high QE, what is QE? The equilibrium absorption capacity. So with a high equilibrium adsorption capacity will typically show a higher rate constant. Okay. So what we can conclude is that nanomaterials with a larger surface area, abundant active sites, and strong binding affinity will exhibit a faster removal process, and it will show a higher efficiency. So all these factors, these are involved in the faster dye removal process and for the higher efficiency of your catalyst. Thus, adsorption capacity provides insight into the catalytic effectiveness. Nanotechnology for pesticide removal. So pesticide is such a huge menace, as you already know. The famous book by Rachel Carson that was published in 1962, that was Silent Spring, which created an entire revolution for banning the toxic pesticides. It showed the harmful effects of the pesticide DDT that was used very popularly, that was very popular back then. So a lot of chemical industries, they revolted against that book and they banned that book. But slowly people realized that that book was right, and DDT has created a huge menace, and it has entered into the food chain, and it was causing so many illnesses, and it was very highly toxic. So that resulted in the ban of DDT, and also authorities were created to regulate the pesticides that are being used. So now pesticides that are being used are very highly regulated. You cannot just use any pesticide because they are highly toxic. So the certain pesticides, they have already percolated through the environment, and they are already persistent in our water bodies. So these have contaminated and they stay there. So nanomaterials will basically, they will adsorb and degrade pesticide molecules. So nanomaterials have provided a sustainable solution to this problem. So they can adsorb and degrade these pesticide molecules. So photocatalytic nanomaterials, basically they break down complex structures, and they can do that very efficiently as compared to your conventional adsorbents that are being currently used. Then removal of pharmaceutical residues. So pharmaceuticals are also emerging water pollutants. So nanoadsorbents can capture antibiotics and hormones as well in a similar fashion. So catalytic nanomaterials can degrade active compounds, and they can also reduce ecological and health risks. The removal of microbial contaminants. So bacteria and viruses also cause waterborne diseases, like I've already told you, and the membrane, you've been using membrane for water purification. Those can result in fouling, and they can be highly expensive. So we need some sustainable and cost-effective solution to this. So silver and metal oxide nanoparticles exhibit antimicrobial action. Nano-enabled membranes will enhance the filtration process. And then disruption of microbial cell walls and DNA. What will your nanomaterials do? These can also cause disruption of the microbial cell walls and DNA and render them ineffective. So that's the virus detection technique. That's a single virus detection using nanowires. On the left you can see a schematic depicts the two nanowire devices, that is 1 and 2. That's a unique antibody receptor attached to this. Only nanowire 2 does a single virus's precise contact. What will happen with the 2 is that a single virus's precise contact with a receptor will cause a conductance change. See, a conductance change will be observed over here on the chip. So a single virus comes and attaches itself with this antibody, and your conduction will show a change over here. And then that's the virus will cause a conductance change, and that is the sign of the virus's surface charge. So following the virus is released from the surface, when the virus is released from the surface, the conductance value will return to its original value. So that is how you can detect it using a nanowire. Then for mechanisms of microbial inactivation, there are generation of reactive oxygen species. Like I've already told you, nanocatalysts, they generate reactive oxygen species and which directly interact with the cell membranes of your microbes, and they can enter inside the cell membrane of your microbes, your bacteria and viruses, and they can cause protein and DNA damage. Okay, they can damage their genetic materials, or they can and they also inhibit the metabolic pathways via this reactive oxygen species that they generate. They can also interact with the cellular membranes. This is how they inactivate microbes. That's an elaborate diagram of, see, nanoparticles. These can cause membrane damage. And they can inhibit the electron transport chain. They can damage the proton efflux pump. They can be releasing this. They can damage the membrane, and cause the generation of reactive oxygen species. These reactive oxygen species, what will they do? They will integrate with the DNA and damage it. Okay, these can intercalate and damage your DNA. These can cause protein damage or turn them into some toxic products, and they can also cause enzyme disruption, and all this can lead to cell death. Bacterial or fungal cell, whatever cell this is, that is how your nanomaterial is going to damage your microbe. Then it can also cause mitochondrial dysfunction or destabilize ribosome. So all these are various methods by which your nanoparticle is going to disrupt your bacteria, and these also prevent biofilm formation by attaching to the membrane. Then let's discuss some representative case study examples. Let's study what the photocatalytic degradation of dyes is like, and then removal of pesticides, then antibiotic adsorption by carbon nanomaterials, or bacterial inactivation using silver nanoparticles. Let's discuss these cases one by one. So that's a picture from a research article where the possible photocatalytic reaction mechanism for methylene blue dye is being discussed by silver oxide, zinc oxide nanoparticles. So that's your nanocomposite, and what is happening is that the same way the radiation is falling onto it, and your electron is getting excited from valence band to conduction band, and then that will break down your water and into a hydroxyl anion and hydroxyl radical, and these will go on to degrade your methylene blue dye, and that will result in the release of non-toxic degradation products. So that will your methylene blue dye will be broken down into small organic molecules that are not that toxic. That's the schematic illustration of the primary antibacterial mechanisms of silver oxide. So silver oxide, as you already know, they are already used in antimicrobial ointments or ointments for used for burns and all. So you already know they are highly being used, and they are very antibacterial in nature. So that's the mechanism. That's the step 1, where silver oxide nanoparticles come and adhere to the microbial cell surface. They are coming and sticking to the cell surface, causing membrane disruption and altering transport functions. Then number 2, these are passing through the membrane, penetrating the microbial cells and interacting with the intracellular components and disturbing some important cellular activities. Then 3, what is happening is that your silver oxide nanoparticles are producing the formation of reactive oxygen species. These will be interacting with your biomolecules and forming reactive oxygen species, leading to oxidative stress and cellular damage. And then fourth, that your nanoparticles are interfering with microbial signaling pathways, triggering programmed cell death. Okay, these are now interfering with your microbial signaling pathways and triggering the programmed cell death. And then fifth, these are disrupting ion transport across the microbial membrane, further impairing essential cellular functions. So this is how your that is your S. aureus, your bacteria on which your silver oxide nanoparticle is working in these steps. So this is how you target a microbe using a nanomaterial. And these are the processes via which your nanomaterial is going to kill your microbe. That's photocatalysis of pesticides irradiated by visible or ultraviolet light. So that's your light or UV source, and the same process is happening. Your electron is getting excited from valence band, it is jumping to your conduction band, and then that's the electron and that's the whole pair. And what is happening is that your superoxide oxygen radical is being created and your hydroxyl radical is being created, and these will go on to degrade the pesticides in water. So these are mostly organic compounds, and these will be broken down into some non-toxic products, especially H2O and CO2. So that is how pesticides are being degraded. Then adsorptive removal of antibiotics from water. So you can use some carbon-based materials like graphene, carbon nanotubes, biochar and activated carbon for the adsorptive removal of antibiotics. So antibiotics are coming and absorbing onto a surface, and you are removing it. So basically the size range you are targeting, that is 1 to 100 nanometer. That's a clear picture of antibiotic removal. So that's an experimental picture where carbon nanotubes have been used for your antibiotic removal. So you can see the carbon nanotubes, they have a very high surface area, and these have these spaces, and what will happen is that your antibiotics are being removed via various processes. Your antibody can interact with this nanomaterial via hydrogen bonds. See, these can interact with these via hydrogen bond. That's a section of this carbon nanotube. So these your antibiotics are basically forming hydrogen bonds or surface precipitation, pi-pi interactions. This is how they are going to come and absorb, or hydrophobic interaction and non-carbonized fractions, or pore filling. They can get inside the pores and get absorbed, or electrostatic attraction. So they just get adsorbed onto the surface and removed. So the high surface area of the carbon nanotube and the porosity will help in enhancing the adsorption effect, and it will help in the adsorption and hence removal of the antibiotics. These are very relevant to nanobiotechnology. Bio-inspired nanomaterials are used for water purification and integration of biological and nanotechnological approaches. You are integrating biological and nanotechnological approaches, and then eco-friendly and sustainable treatment strategies. These are offering an eco-friendly and sustainable treatment strategies and alignment with green chemistry principles. Summarizing the entire lecture, nanotechnology has enabled advanced water treatment solutions, and kinetic analysis is essential for the performance evaluation. So you are using a nanomaterial, you need to make a kinetic analysis of it, whether it is performing as a good nanocatalyst or not. So for performance evaluation, you need to do a thorough kinetic analysis of your material that you are considering. Then effective removal of dyes pesticides, pharmaceuticals and microbes. Effective removal of dyes, we have studied this. That is the effective removal of dye, methylene blue, effective removal of microbes by antibacterial mechanisms. That's the effective removal of pesticides by photocatalysis. That's the adsorptive removal of antibiotics from water. That is the adsorptive removal of antibiotics from water. So all these are already taken from research papers, which means these are already in study. So you can see that the removal of dyes and pesticides in pharmaceuticals and microbes are very effective via this nanomaterial. When we use nanomaterials, there's an effective removal of dyes, pesticides and pharmaceuticals and microbes, and this plays a central role in sustainable environmental remediation. Thank you.
Hi, I'm Chitwan Lalji, a PhD student of Health Economics under the supervision of Dr. Debayan Pakrashi from the Department of Humanities and Social Sciences, IIT Kanpur. In one of my essays, I'm interested in understanding the relationship between consumption of fruits and vegetables and various health indicators. Health indicators, both subjective and objective health indicators like mental health, self-assessed health, various measures of blood pressure and various measures of cholesterol. Measures of blood pressure like systolic and diastolic BP. You have your incidence of high BP, MAP and incidence of high MAP. And as far as cholesterol is concerned, I have tried to concentrate more on total cholesterol, good cholesterol and incidence of high cholesterol. Now before I go on to what have been my major contributions and various policy implications, I would like to briefly tell you about the policy initiatives of WHO and various countries. The WHO, that is the World Health Organization, it started with a campaign of 5 a day. That is, you should have 5 portions of fruits and vegetables per day. That would be approximately, you could say, 400 grams of fruits and vegetables. Now, a portion, before we go further, I'll just tell you what exactly is a portion. One portion is equivalent to a medium-sized apple, or one small glass of fruit juice, which is approximately 150 milliliters, and maybe 3 teaspoons of vegetables. So, the WHO went with a 5 a day campaign, which was further taken up by various countries. Countries like UK, Netherlands, Germany, Norway, they adopted the 5 a day policy, while some went for expansionary dietary policies like France, Australia, Canada, Denmark. So, for example, Australia, it went for 2+5 policy, in which it said that you should consume 2 portions of fruits and 5 portions of vegetables per day. And USA went for a policy of fruits and vegetables more matters. That is, you must consume more and more fruits and vegetables. Now, irrespective of these expansionary dietary policies and dietary propagations, it has been found that only 28% of women and 25% of men, they actually meet the recommended dietary norms of 5 a day portion. So, the major contribution of my work is firstly to find an association between fruits and vegetables, whether there exists a relationship between fruits and vegetables and health indicators, and if they exist, whether due to heterogeneity in the data. So, I will be doing it according to age, by gender and by your weight. So, apart from that, I will go for policy recommendations, in which I am basically studying how much fruits and vegetables matter. Apart from that, which type matters more. So, for that, I have taken data from the Health Survey of England. Health Survey of England is an annual survey which takes data, which conducts information regularly on demographic and socioeconomic characteristics. You have your lifestyle behaviors like an individual smokes or doesn't smoke, alcohol consumption, you have your sedentary and physical activities, and you have various health indicators also which have been collected. Before I go on to what exactly is my research, I would like to concentrate more on fruits and vegetables, like what kind of questions were asked in the survey. Questions like, what kind of fresh fruit do you eat? did you eat any dried fruit yesterday? Don't count dried fruits in cereals, cakes. Apart from that, for vegetables, they asked how many tablespoons of vegetables did you eat yesterday. So approximately after this whole survey was conducted, data was converted into portions of fruits, and like for example, 3 tablespoons of vegetables is equivalent to 1 portion. So data was converted and provided to the users, that is us, from the UK Data Health Survey. So the major contributions of my paper is that I found a strong negative association between intake of fruits and self-assessed health, then various measures of blood pressure like mean arterial pressure, high blood pressure, systolic and diastolic BP, and your total cholesterol. Apart from that, I have found a strong positive association between consumption of vegetables and good cholesterol. So it is recommended in a way that if you want to control your blood pressure, you must consume more and more fruits. And as far as vegetables are concerned, they impact your good cholesterol. Apart from that, I went in for a falsification test. A falsification test is basically conducted to know whether the model that you have adopted and the conclusions that you are drawing are not spurious. So, if a falsification test is done to know, in a way it is tested by seeing an indicator, a health indicator which is not being impacted by your consumption of fruits and vegetables, and then we see whether there is significant result or not. If there is no significant result, that means your model is good and your results are non-spurious. So what we did is, for falsification test, we took ear complaints and infectious diseases. Now ear complaints, like if you are deaf since birth or you have some kind of imbalance, body imbalance, that is not being impacted by your consumption of fruits and vegetables, and we did find insignificant results. Apart from that, infectious diseases like HIV, AIDS, etc., we found similar insignificant results, indicating that our results are not non-spurious. Apart from that, we went, since there was a lot of heterogeneity in the data, like by gender, by age and by weight, we did the regression analysis. We found results which stated that as far as fruits are concerned, it impacts a male's health more than a female's health. So it is basically said a man should consume more fruits to impact his health, whereas as far as vegetables are concerned, they impact a woman's health more. But this has been only seen as far as cholesterol is concerned, the various measures of cholesterol like total cholesterol, good cholesterol and your incidence of getting high cholesterol. Now after this, we went in for a policy implication, and in the policy implication we tried to find 2 policy implications: what matters and exactly how much portion matters. So as far as how much portion matters, we have found that on an average 5 or more portions of fruits impact your overall health, that is your self-assessed health, your MAP, your incidence of high MAP and incidence of high BP. But if you want to have a good mental health, so you can optimize your mental health by consuming 3 or 4 portions of fruits as well. And similarly, as far as self-assessed health and total cholesterol is concerned, an individual must consume 4 to 5 portions to optimally have the impact of consumption of fruits. Apart from that, vegetables have had a very little impact on your health. It only impacts your incidence of getting high MAP and high BP, and it's seen that only it impacts when you consume 5 or more portions of fruits. So an optimum consumption of 5 or more portions of fruits and vegetables are recommended, but fruits have a more impact on your overall health, on various measures like self-assessed health, mental health, your various measures of blood pressure and various cholesterol levels. Another thing that we find is which type of fruit matters. It has been seen that all size fruits, they impact your self-assessed health, your systolic and diastolic blood pressure, your mean arterial pressure, your high BP and incidence of getting high MAP and high cholesterol. But we find that as far as frozen fruits or canned fruits are concerned, they help in regulating your incidence of getting high MAP or high BP, but it has a trade-off. That means there is something negative happening. It reduces the good cholesterol in your body. Apart from this, if you have an incidence of getting high cholesterol, it is recommended that you must consume fruit juices, because it has a impact in reducing your probability of getting high cholesterol. and dried fruits, they impact your self-assessed health. As far as vegetables are concerned, very little impact has been seen. It has only been seen in case of portion of salads and its association with self-assessed health. Another thing that they have seen is vegetables in composite, they have an association with good cholesterol. So overall, my research basically says that there is an association between consumption of fruits and vegetables and various health indicators, and it is highly recommended that an individual, in order to be healthy, must consume 5 or more portions of fruits and 5 or more portions of vegetables per day. But fruits have a more impact on your overall health. Apart from that, all size fruits, they have a better impact on your overall health, your mental health, various measures of blood pressure and cholesterol. So, thank you.